We have Lisa Sercombe, our product manager, based in Melbourne, who will give the first talk. And then we've got Yvonne Peterman, the science and technical advisor from 10x Genomics, who's also based in Melbourne, doing the second talk. So I think what we'll do is get going, and at the end of Lisa's talk, we can have some questions, and then at the end of Yvonne's, we can have some more. But yeah, feel free to put more in the chat and along the way as well. Right, over to you, Lisa.
Thanks. I will share my screen. So today I'll be providing some recommendations and advice on common laboratory workflows to investigate cellular metabolism. Okay, so just a quick outline of what I'm going to talk about today. I'm going to give some background on metabolism, some considerations for metabolic measurements, as well as describe some various measurements that you can utilize in the laboratory with the Agilent Biotech range. I'm also going to go through 3 common examples of data for evaluating metabolism. So cellular metabolism in its broadest definition is a series of chemical reactions to maintain life. And it's comprised of catabolic and anabolic reactions that are linked together in a pathway via enzymes within the pathway, as well as nutrient availability. An image, first one here, is often what we think of when metabolism comes to mind, but the reality is far more complex. And so the second image here is a more accurate representation of metabolism. When it comes to metabolic experiments, there's a wide variety of targets that you can look at and options. You can measure the rates of change of metabolites or the metabolites directly, the reactants and products in cellular respiration, as well as building blocks as well. When we look at cancer, in and of itself, cancer is a heterogeneous and complex collection of diseases. And yet, despite this heterogeneity, there are a series of fundamental hallmarks that are shared within tumours. And an example of that is altered metabolism. And this can be exploited clinically. So under normal conditions, in the presence of oxygen, tissues will metabolize glucose into pyruvate via glycolysis, and then oxidize a large fraction of the pyruvate to CO2 via oxidative phosphorylation. Under anaerobic conditions, normal cells will redirect pyruvate away from mitochondrial oxidation, generating lactate. But in cancer cells, they tend to rely on aerobic glycolysis, which leads to the accumulation and use of intermediates in the production of macromolecules required for proliferation, as well as reactive oxygen species. So when it comes to metabolic experiments, there are a few general considerations. These include your media components. Do you need the cells to be serum starved? What concentration of glucose do you need? As well as location and timing. So what point in which are your metabolites being produced? Do you in fact need to measure your metabolites at multiple time points, or perhaps even look at multiple metabolites themselves? Plating density will affect all manner of cell behavior. So do you need proliferating cells or cell-to-cell contact? As well, the more cells that you're plating, the more glucose that is being consumed and pyruvate being produced. Sensitivity or lack thereof. So does the readout accurately reflect the research question in which you're trying to address? And remember that different methods will produce different sensitivities, from plate reading to imaging to mass spec. And lastly, your live cell reagents and artifacts. So what is the time in which your components are being used and background can be produced? As well as if you're using cytotoxic reagents, these can affect viability and, as a result, metabolism. So there are several options which you can use to normalize your metabolic data. From plate reading, you can look at total protein through a absorbance-based BCA assay, DNA quantification, and with biotech, we have the Take3 plate, which allows you to do high throughput microvolume analysis, as well as your luminescence assays that you can utilize, such as cell titer glow. There's also imaging options you can utilize as well, from cell count, viability assays, and mitochondrial morphology. Now, cell count is an optimal option for normalizing your data, and this image here really does illustrate this beautifully. So when looking at detection, we can see the cells of interest. We see with the reality of imaging, we can see that there are some cells that are washed away, and also background through media and dye. So by combining the cell count with your data, you can get the most accurate picture for your information. So the global solution for capturing your metabolic data, including what was just shown on the slide previously, is the Citation 5 from biotech itself. So it's a multi-mode plate reader with sophisticated imaging. And the plate reading component is the hybrid optics, which biotech is known for. So it utilizes a monochromator and filter-based optics for flexibility and performance. And the fluorescence-based monochromators use variable bandwidth, which allow you to have sensitivity and specificity. The imaging component has the advanced microscopy, which really does allow you to have unlimited possibilities for your data. And an example is what we can see here through heat picking. So through the combination of multi-mode detection, you can focus down into your areas of interest and simply capture those wells, which allows you to save time and data storage. When it comes to mitochondrial measurements, there are a whole host of options which you can utilize in the lab, from measuring ATP, membrane potential, as well as your typical cell viability or proliferation assays, such as MTT. And this measures the activity of a mitochondrial enzyme. So it's something to keep in mind as well. On this slide here, we have a wonderful visual abstract that is illustrating with the biotech range. So in this slide here, we are measuring the extracellular oxygen consumption with a fluorescent biosensor. So oxygen consumption is a metric that can be used to determine the effects of treatments on cellular respiration. And one way to measure this is to look at how much oxygen in the microenvironment is being expended over time. An example of one such kit is the Mito Express Extra Oxygen Consumption Assay from Agilent. We have this image here, which it illustrates the mechanism of action. So this assay uses time-resolved fluorescence, and this allows you, with the probe signal sensitivity, it's enhanced by acquiring data following 2 different emission delay times over the fluorescent curve. So using the software itself, you can define these 2 steps. You can then detect the change in extracellular oxygen levels for various cell treatments, and this is measured kinetically using both of those read steps defined earlier. From this data here in the software, you can calculate a ratiometric lifetime value from the values of each read step. And the oxygen consumption rate is computed as the slope of the linear response portion. And finally, you have your reported data. So compared to your untreated cells shown in purple, an uncoupling reagent shown in the 2 blue bars drives maximum oxygen consumptions, whereas use of electron transport chain distributors, which we can see in red and green, inhibit oxygen consumption rate. So an example of another option from the biotech plate reading capacity for capturing metabolic data is the Synergy Neo2, and it's one of the instruments capable of detecting the data shown in the slide previously. The Synergy Neo 2 is our flagship plate reader, and it utilizes, similar to the Citation 5, the hybrid optics. It has independent optimized optical paths, same like the Citation 5, the variable bandwidth for sensitivity and specificity. Another features which really does drive excellent performance for your TRF assays and ratiometrics is through the use of dual PMTs. And this allows you to look at two measurements separately, but at the same time. And the Synergy Neo 2 can also be built in with a TRF laser for nice clean data, and we can see this with the Z prime. So the Neo 2 really does allow you to have uncompromised speed and performance for your cell-based and biochemical assays. Another example of some data you can utilize for detection to look at metabolism is the Mito-ID membrane potential cytotoxicity kit. And this is from Enzo Life Sciences. It's an alternative to the widely used JC1 kit. And JC1 does have some challenges as it's poorly water soluble and requires multiple wash steps. On the other hand, Mito-ID has been engineered to be more water soluble, and so it doesn't require the wash steps or medium removal. It's also allows for live cell staining. It's 10 times more sensitive. It's also a photostable dye. So how it works is that the dye itself enters selectively into the mitochondria, and as the membrane potential increases, the dye reversibly changes from green to orange. So low membrane polarization is green, and as we have the mitochondrial polarization, we change from a orange to red signal. Here we have some data here. So some HeLa cells were treated with CCP and the Mito dye, or the JC1 dye was used in a microplate reader. And what we can see that MPP was shown to decrease using the CPP increasing CCP concentration. And you can really see here that thanks to the improved aqueous solubility of the Mito-ID dye, we do have minimized variability thanks to a higher Z factor. Another option that you can see here, thanks to the Mito-ID, it's really a fantastic kit that allows you to look at membrane potential in real time. When it comes to metabolic stress, an option that you can utilize is autophagy measurements. So autophagy is a degradation and recycling pathway that allows you to adapt to undesirable conditions. And whilst it's used to activate and promote cell survival, it can progress to cell death. And there are a number of options available to examine autophagy. One option is the Cyto-ID autophagy detection kit, and this is also available from Enzo Life Sciences. So the probe is a cationic amphilic tracer dye that rapidly partitions into cells to induce phospholipidosis. An advantage of this kit is that no transfection is required, so it will save you a significant amount of time. It's specific as it differentiates between autophagic flux and autophagolysosome accumulation with negligible background, along with a whole host of additional features. We have an example of data here for detecting autophagy with this kit. So Hep G2 cells were treated with tamoxifen for 16 hours and then treated with the Cyto-ID dye. And what we can see here is with increase in Cyto-ID autophagy dye signal in the cells at concentrations greater than 1 micromolar. We use Veprimil, another treatment, which passively diffuses into lysosomes and becomes trapped upon pronation, causing an increase in lysosomal pH. And so this inhibits lysosomal function and blocks fusion with the autophagosome. So what we can see in this data here is that at concentrations of Veprimil greater than 10 micromolar or greater, we can see an increase in the Cyto-ID autophagy dye signal. Okay, so in summary, metabolic changes represent a broad but comprehensive tool to study cancer as well as cellular biology in general. And there's a diverse range of metabolic targets that we can use to evaluate metabolism and also exploit clinically. And there's a variety of strategies to detect these targets of interest. Plate readers, especially the biotech range, it offers a rather inexpensive but diverse technology to comprehensively examine your research questions. Excellent. Well, thank you very much for listening. Please let me know if you have any questions. I'll be happy to answer them for you. How does the data compare with what you can see from the seahorse? It really depends on what you want to look at. The seahorse is another option that is capable to look at your metabolic data. And Biotech and Agilent have actually produced some information about able to integrate the citation one with the seahorse itself. And we recently, I think Cameron, our imaging specialist, recently helped with an integration. So it really depends on your area of interest. And we have some tech notes that we can share to you if you'd like as well.
So yeah, good morning everyone, or good afternoon. My name is Yvonne Peterman. I'm a science and technology advisor, and with today's presentation, I want to give you an overview of all the single cell and spatial multiomics solutions that we have, so that our range is ever expanding. So I'll try to be as brief but as specific as possible, and I'll also share some examples across the talk. And as mentioned before, please feel free to pop in any questions as we go into the chat box, so we can answer them in the Q&A session afterwards. So I want to begin with an overview of the 3 platforms that we'll soon have, with Xenium being launched early next year. But currently in the market, we have our Chromium single cell solution with the Chromium instruments, and our Visium spatial transcriptomic platform or solution that's being done on the Visium slides. So you might all be familiar with these two systems already. And both of them, depending on how many cells you're running, but it is allowing you to conduct complete or total discovery of new biomarkers, cell types, rare cell types, rare transcripts, etc. And as you move into the Xenium platform, which is going to be our sort of highest resolution platform, enabling translational and protein expression analysis of more clinical applications, really then takes you into the very focused approach. And our Chromium single cell solution here, you see the small Chromium controller instrument that enables the droplet generation or generating the gems, gel bead and emulsions. You can really run these 4 different assay types on this instrument, loading it on this microphage chip, and I'll talk a little bit more about it. And in the center here, we see a schematic of the Visium spatial gene expression slide, where you have the opportunity to look at 4 tissue sections in one experiment. And this is now compatible with both fresh frozen as well as FFPE tissue types. And the list of validated tissues that we've tested on both of these workflows is ever expanding. So it's most likely that if you are considering to do spatial gene expression analysis with these slides, we might have a validated protocol available. So what we see is an ever increasing list of publications, and I like showing the slide because I started a year ago with 10x and we were sitting at 2,000 publications then. So it's really impressive how the list of publication increases, but it's not really just about the numbers of papers, it's also their impact and breadth of what's being shown, of what's being discovered, and how in all areas across oncology, immunology, neuroscience, etc., how this implementation of the technology really advances the understanding of many research fields. And some of the answers that can be revealed using single cell gene expression, are sort of listed here. You can unlock answers to specific cell types, lineage function. You will be able to better understand of how cells contribute to tissue and organ biology, and hitting on all modalities that contribute to a successful target identification, validation, preclinical and clinical studies for cancer are really enabled. So there is, lots of different applications available here where we see cell atlasing being done, looking at tumor heterogeneity, identifying rare cell detection, and elucidating mechanisms of cellular development. And that's just really naming a few. The applications are really very varied and vast. So I want to begin with the Chromium single cell workflow, and just going over the workflow steps at high level. You would have seen this probably many times by now, where you can apply these 4 different assay types. It is a robust, ready-to-use workflow once installed in the lab. We have a lot of demonstrated protocols available for lots of different sample types. Typically, you do put in cell suspension, and there's many different ways of generating the cell suspension depending on what your starting material is, whether it's tissue, whether it's frozen or fresh, whether it's blood or any other body fluids, or whether it's cell cultures or organoid cultures, etc. So for all of these different input types, we have methods available to generate these highly viable, clean cell suspensions that you then load onto the Chromium controller microfluidic chip along with one of these assays. And within a 20-minute run on the instrument, we do generate these individual droplets capturing these individual cells. And with a number of steps that you conduct on the bench, you do generate sequencing-ready libraries that will be sequenced on Illumina sequences, which is the one part that 10x doesn't have an involvement with, but we do provide the analysis and visualization software tools for free, along with lots of demonstration videos and sample data sets. But you are not locked into using these. You can also take these common output files that Cell Ranger, for example, produces, and you can use a third-party community analysis tool to look at your data. The same with visualization. Loop Browser is a fantastic tool. It is fairly easy to use. It does have some limitations. So if you do need to go beyond what Loop Browser is capable of, you can also plug in some of the output files from Cell Ranger into these community tools. So here, this is a bit of a closer up graphic of how the 10x microfluidic technology works, beginning with the 10x barcoded gel beads, which you'll put in millions of those, and then you load your cell and enzyme mix onto the microfluidic chip, and the aqueous phase will be combined with the oil, and then we generate these droplets side by side within the instrument within a short 20-minute run. And then you collect that emulsion that you've generated, and the majority of cells of droplets will contain an individual cell or an individual nuclei, depending on what you've put in. But we'll also have a number that is empty, and we'll have a number that have multiple cells and nuclei captured. But once these gems have been generated, those gel beads melt and the cells lyse, and so the biochemical reaction will start occurring. We do reverse transcript all the captured mRNAs, and we barcode the resulting cDNA is barcoded and then can be pooled, and the final library preparation can occur on the bench in bulk before you then load this on the sequencer. And how capturing transcripts and additional analytes is facilitated is shown here on this graphic of what our gel beads look like. And I don't want to go too much into detail, just highlighting that we do capture polyadenylated mRNA only with the platform. So just keep that in mind in case you are interested in, for example, looking at bacterial RNA that is not compatible with this platform. And we do have included additional oligos carrying a capture sequence 1 and capture sequence 2, and via those we facilitate the capturing of additional analytes, which is what our multiomics or what is enabling our multiomics. So apart from detecting the whole transcriptome, so really measuring the digital gene expression from every single cell, these additional oligos are present on the beads, allow you to, for example, study cell surface protein. This is conducted by utilizing antibodies that are tagged with a small oligo, which sequence is compatible to the capture sequence 1 or capture sequence 2 on the bead, and BioLegend has ready to use antibody pools. That's their total seq-B portfolio of antibodies that can be applied, and you can really look up to even more than 100 cell surface proteins. So there is, from the technology point of view, no real limit, but of course it is a cost factor when you consider purchasing these large antibody panels, but they can also be customized. You can just pick a handful and order those in as well. And lastly, with the 3' gene expression solution, we have enabled CRISPR perturbation screening, so we can profile the whole transcriptome or the targeted genes specifically, but also detect the single guide RNA from your CRISPR screen that's expressed in each individual cell. So really, if you combine all those, you're basically getting 3 different readouts from every single captured cell on the Chromium controller. And with this image here, I wanted to show you what some of this data looks like. Here specifically on the example of a colorectal cancer sample where we've profiled 8,370 cells to study and really dissect the tumor microenvironment. And it's shown here on a so-called t-SNE plot, which is really a way bioinformatically of showcasing this high-dimensional data in this 2-dimensional space, where each individual dot represents an individual cell, and cells that are plotted very close together have a very similar transcriptomic profile, and cells that are plotted, at a much greater distance have a very different transcriptional profile. So showing you here at high level, we identifying, specific immune cells. We are detecting different types of tissue cells that are present, and we can also detect the tumor cells. And what we also see is with this relatively high number of cells that we are running, that we can actually detect a large number of very specific subcells, subtypes of immune cells. So we can really see different types of T helper cells, for example, as well as in the B cell group, we can see that we have activated B cells, for example, present. So all this gives you insight into the behavior of the tumor, and potentially what you can derive from that information is potentially targeting treatment options for this particular patient. Then there's a couple more examples from, some of the recent literature that has been published utilizing, the groups utilizing the chromium single cell 3' gene expression kit to investigate tumor-associated macrophages, for example, here in glioma biopsies. And this is just one example of the 3' gene expression kit or its application. This is done here by the University of California in San Francisco to show that to show more detail on this type of glioma, or a glioma, which is a type of tumor that occurs in the brain and in the spinal cord. And what they were able to identify is that these blood-derived tumor-associated macrophages are infiltrating these gliomas preferably or preferentially, and preferentially express immunosuppressive cytokines. This one here is an example of looking at gastric samples or gastric cancer samples. In the center here, you can see the study design, or what types of samples have been collected at what stage and at what location. And the group here, Zhang et al., has also used the chromium single cell gene expression solution and constructed a cell atlas. So we are seeing here on the right the result of the profiling of 32,000 or just over 32,000 cells. And we can see all the different types of cells that have been identified. But what was very exciting for these, for the authors here specifically, that they could identify high confidence genetic markers of potential clinical utility as diagnostic markers in early stage cancer. So this group here had previously done a bulk RNA sequencing, and they failed with that method to identify or distinguish these specific cell types, and they weren't able to detect rare cellular populations within these specific states that they were looking at. So really the 3' gene expression solution gave them that fine-tuned information by also, allowing them to study that many cells from different cancer patients. And this is a very recent publication from Jambando et al. Just came out in stem cell. They have used here this 3' gene expression solution as well on cardiac fibroblasts in mouse samples. They've FACS sorted these mouse cardiac fibroblasts and applied those specifically. They actually did a range of different types of analysis. So this is just a little snapshot of what they found using the chromium single cell gene expression. And they were able to really identify the HIF1A function within this cardiac fibroblast proliferation, and how this influences the myocardial infarction in mice. So if you want to look at that in further detail, I've put the DOI at the bottom of the slide. So next up is the immune profiling solution. Here we are looking at the 5' transcript, or 5' end of the transcript, compared to our 3' gene expression solution, as the name convention already gives away. So really we are doing this because at that end we are able to capture the full-length VDJ sequences from your cells. So here you basically are able to simultaneously profile paired and full-length T and B cell receptor sequences at the same time, and really get that clonality information in your sample. So just using the similar graphic that I used before in this 3' gene expression solution, we have also enabled several multi-omics approaches with this kit as well. So again, you are able to profile the whole transcriptome. You can isolate paired immune receptor sequences, but you can also look at the cell surface proteins again. Very similar mechanism as the 3' gene expression solution. It's just a different product range from BioLegend for total CC, antibodies here that are already compatible with 10x. And you are also able to look at antigen recognition by using specific labeled antigens that you can also purchase from Immunodex, for example. Just giving you a quick snapshot of what this looks like when we dissect a sample on in Loop browser. So you see 5' gene expression again, and sometimes we get that question. There really isn't much of a difference in the gene expression we are capturing from the 5' or the 3' end with the two versions that we have out there in the market. They're really both very, very comparable. But here, in addition, we have run a panel of specific antibodies to really confidently detect specific cell types like the macrophages, monocytes, NK cells, etc. So you see these clustered here on the left, and then on the right, you can see the ability that you have with Loop browser of, for example, looking specifically at the B-cell cluster and picking out the different immune globulin clonotypes that you are obtaining with this immune profiling kit. And similarly, if you look at the T-cell receptor or the T-cell cluster here, the orange cluster, you can then zoom in and look at the T-cell receptor clonotypes in much greater detail. And this here is done on PBMCs. And you can also see, you know, as a high-level heat map overview from the PBMCs, the different fold changes in their different expression levels. This is here a great example of making use of this 5' immune profiling solution on just studying transcriptional mechanisms of immunotherapy resistance. In this publication here, the authors have used samples derived from patients with metastatic Merkel cell carcinoma, and they've treated these patients with these autologous Merkel cell polyomavirus specific CD8 positive T cells that have been specifically engineered to recognize a key onc protein for this tumor type and immune checkpoint inhibitors. And in both cases, in both patients, the patients went into remission, and so the tumor regressed. However, both patients actually went into relapse several months later. So the authors really wanted to understand what occurred in these particular patients, because what's been published previously is that this resistance occurs because the treatment then also didn't work on these patients anymore. And what the publications, previous publications suggested is that the resistance is most likely due to the loss of these CD8 positive T cells that are supposed to initiate the response. However, the deep analysis using the immune profiling solution actually shows that they're still present. However, what's being shown here is that the tumor actually transcriptionally shut off HLA. And so although these T cells were there and could recognize the antigen, it could no longer be presented. So that was a particularly interesting finding, and also provided a little glimpse of hope, because this pathway specifically, this transcriptional HLA repression, is known to be reversible. So the authors then went ahead to work out on the pathway to reactivate that for these patients. I want to now touch on these two epigenetic analysis options that we have with our platform, which are our ATAC-seq and our multi-omic assay. And just so I'm not going too much over time, I might skip this essay of this slide of why is it so exciting to look at the epigenome. So you can look at specific drivers, transcription factors, so drivers of differential gene expression, understand cellular diversity and characterize developmental trajectories and cellular responses by just using the open chromatin. So we do this with the assay for transposase accessible chromatin, which you can get as a standalone assay using a nuclei as an input. But you can also try the multi-ohm kit that captures the accessible chromatin as well as the transcriptome from the single cell by utilizing nuclei. So two really powerful kits that give you some, deep understanding of, in this example here, of PBMCs. We are looking at, at the top there, some bulk ATAC-seq, but then how much more detail you can achieve using the single cell ATAC-seq kit. And I might just briefly skip over this so I can cover some more spatial gene expression applications in the last 5 minutes of the talk. We have here an example of an implementation of this ATAC-seq kit and how you can basically use the single cell chromatin landscape to study the immune cell development and intratumoral T-cell exhaustion in your samples by utilizing nuclei. In this case here, it's derived from bone marrow blood and basal cell carcinoma cells. So I want to spend 5 minutes on this Visium spatial gene expression because I think this is another fantastic technology that allows you to really interrogate gene expression, but in the spatial context, because location does matter in a lot of cases, which is shown here on this particular example, where we are looking at a H&E stained image here, a tumor stained image, and we are looking at tumor infiltrating lymphocytes that are sort of depicted here in this pink color. And at the top, the spatial analysis reveals that these infiltrating lymphocytes have infiltrated the tumor. And this is considered being a hot tumor with a good prognosis, good outcome for this particular patient. Whereas at the bottom, we see again in the tumor, looking at tumor infiltrating lymphocytes on the right, that they have been somehow held back. So they're not infiltrating the tumor, which is considered a cold tumor and resulting in poor prognosis. So for cancer analysis, just simply looking at the spatial transcriptomics, you get additional information from your samples that you don't get at the single cell level. Why this is also exciting is because this doesn't require a specific 10x instrument. The magic happens on our Visium gene expression slides, which you can see here on the left. However, you will need a histology capability to prepare these sections, either cryosections or FFPE sections, and you will need to have capability of taking images of these sections, either bright field or immunofluorescence. And again, the same is required. We do need an Illumina sequencer for the library sequencing, and the visualization data analysis tool are available from 10x. So the top here highlights the construction of these Visium slides, that you are able to get a resolution of 1 to 5 cells per spot, can be 10 cells per spot depending on the tissue size. And this really is derived from the dimensions of these little barcoded spots that you see in the center there at the top, which is 55 micrometers. So you're capturing within the spot the transcripts from as many cells as are covering these individual spots. But the good thing is that you capture everything on this section, so you don't have to pick any specific areas of interest. And what we are enabling going forward is we are enabling multiplexing of antibodies, so you can use pools of antibodies on this platform as well. We are doing this in a very similar way to our single cell platform. So we are also using specific oligos that are part of this workflow and that are being detected in our technology. So when this is coming, it will be starting with sort of fairly low plex protein panels. Here in this case, we have a 20 plex protein panel, immuno-oncology protein panel, and we are looking here at an FFPE human tonsil sample, and just showing you how you can look at your protein cluster as well as your RNA expression cluster, and how you can overlay the specific data. So this is going to be really exciting, looking at these different tumor infiltrating immune cells, for example, again, here using a different protein panel on a human breast cancer sample. And again, we are looking at very similar images, and specifically on the right here, you can pick a specific T cell by picking a specific antibody to look at. So you can really look at this in many different ways, picking out the information captured by the many different markers. So this, once it's coming out, is going to be very exciting and applicable to many different sample types and many different sample types, many different immune cell types, for example, depending on which kind of panel you're picking once this is launched. So this is coming very soon and will be allowing you to capture whole transcriptome as well as specific protein expression data at much higher multiplex level, as we have enabled it at the moment with antibodies that are fluorescently labeled. We have also got some larger capture areas coming soon, the middle of this year, where instead of the 4 smaller capture sizes, we will enable larger capture sizes with 11 millimeters by 11 millimeters to really make it much easier. For example, when you look at human brain cancer, human brain tissue sections, where so far you have to do a lot of scoring and running multiple sections. So this is going to also help with the spatial analysis. And a more updated information on the Visium HD, which is expected to launch in late 2022, with single cell high definition resolution here on the example of a mouse eye. So you can see there's barely any gaps left now, and you're really capturing the transcriptome at the single cell level. This here is also an example chosen looking at a breast cancer sample, where you're now able to dissolve the tumor border, for example, in much more granularity than the current Visium solution. So a few more exciting products coming along the way. If one of these on the Chromium single cell platform on the left is of interest to you, please reach out to us so I can provide further detail on some of the improvements that we are making on the ATAC-seq assay. So we are switching out the enzyme there and using the enzyme that's now already used in the multi-omic kit. We will have 5' CRISPR coming, and we also have a fixed RNA profiling kit coming, and a specific nuclei isolation kit as a standalone product as well. So a lot more exciting things to come. But for now, I just want to finish up. Thank you for listening. And we have a few minutes left for discussion or for answering some of your questions. Thank you.
Yeah, if anyone wants to talk about their project individually, yeah, we often have just Zoom chats with Yvonne and I and different customers, and it's sometimes quite nice to talk about the different specifics of your projects, and yeah, just kind of really nail down exactly what you'd need and talk about what you can and can't do. That's really helps some people, put forward proposals or funding proposals, and then so let us know. Our first lunch and learn yesterday with Auckland Genomics, so they have a 10x instrument, but I just thought I'd share this next one that's coming up. Coming up on Wednesday the 23rd. Using whole exome and whole genome sequence and 10x single cell RNA-seq for neoantigen discovery. From 2 researchers up at Auckland. So I think that's going to be really interesting looking at immunotherapy Here's the full abstract if you want to read it. But yeah, basically discussing how they combine those approaches to identify neoantigens from a patient with stage 3 melanoma. Yeah, so we'll send that in the follow-up. We can send the link to register for that. Thank you so much, Lisa and Yvonne, for presenting. And if anyone has questions, I think everyone's got my email, but if not, sales@mscience.co.nz is probably the easiest way to reach me.